We are surrounded by billions and trillions of bacteria, viruses, and other pathogens, and only one of them is sufficient to make us ill and severely ill. Thanks to our immune system that acts as a defense force of our body, saving us from all those harmful pathogens each and every minute and second. Immunoglobulins or antibodies are the specialized soldiers within this defense force trained to recognize and neutralize specific invaders with remarkable precision. Without them, our body becomes vulnerable to countless attacks. Our immune system not just important for our body but also important for our examinations too, as about 2 to 3 questions can be asked from this topic in various competitive examinations like RRB, ESIC, etc. Today, in this video, we will delve into detail of our immune system and their army, that is immunoglobulins, types of immunoglobulins, their functions, and why they are crucial for immunity. So, without further ado, let's start today's discussion. The immune system is a highly complex, dynamic network designed to protect body from harmful invaders. It operates on multiple levels, distinguishing between self and non-self elements to neutralize threats. Key components of our immune system include innate immunity, that's body's first line of defense, offering rapid and non-specific protection through barriers like skin, mucus, etc., and immune cells like phagocytes. Next component is adaptive immunity, that is a specialized system that targets specific pathogens. It involves B cell producing antibodies and T cells that directly attack infected cells. The other component is memory. Once exposed, the immune system remembers the pathogens and enabling faster and stronger responses to future encounters. This synergy between components ensures the body maintains a delicate balance of protection without attacking itself. Firstly, let's look at our types of immunity. Types of immunity includes active and passive immunity, also known as primary and secondary immunity, or innate or adaptive immunity. Active immunity refers to the process of exposing the body to an pathogen to generate an adaptive response. The presence of foreign antigen stimulates the production of specific antibody in the bloodstream, which will recognize and bind tightly to it. This response takes days to weeks to develop, but antibodies last long. These antibodies can be naturally acquired or induced, which is called primary immunity or humoral immune response, where antibodies are produced by B lymphocytes or B cell in body fluids. Vaccination also provides active immunity. Active immunity are long-lasting, often for years or for even a lifetime. It can lead to immunological memory, where the immune system remembers the pathogens and responds faster in future exposure. Passive immunity refers to the process of providing ready-made antibodies to protect against infections. It gives immediate but short-lived protection. They are some form of natural passive immunity, such as maternal antibodies like tetanus antibodies passed across placenta to provide natural immunity for newborn. Artificial passive immunity refers to obtaining serum containing immunoglobulins, for example, anti-tetanus immunoglobulin, human hepatitis B immunoglobulin. However, passive immunity lasts only few weeks or months, as the transferred antibodies are eventually broken down and the body does not produce memory cells. Innate immunity, also called natural or native immunity, provides the early line defense against microbes. It consists the cellular and biochemical defense mechanisms. The mechanisms for innate immunity are specific for structures common to groups of related microbes but may not distinguish fine difference between individual microbes. The principal components of innate immunity are physical and chemical barriers such as epithelial surfaces and microbial chemical produced at the epithelial surface, phagocyte cells that are neutrophils and macrophages and dendritic cells, natural killer NK cells, and other innate lymphoid cells. An important feature of the immune system is immunological memory, which prevents reinfection by same pathogens or antigen when encountered a second time. The immune system reacts faster and more dramatically, producing larger amount of antibodies or immunoglobulins to encounter the antigens when the body is exposed to same pathogen again, which is called secondary immunity. This is mediated by long-lived memory T cells and memory B cells. The cellular response is mediated by T cells. T cells are mainly of two types: killer T cells, that is cytotoxic T cells that recognize and kill invaders directly, and helper T cells that provides essential support for B lymphocytes in the immune response. With completion of this part, you should be able to answer these questions correctly. Do not forget to post your answers in the comment box below. Now, let's start with our second part, that is immunoglobulins. Immunoglobulins or antigens act as a critical part of the immune response by specifically recognizing and binding to particular antigens such as bacteria and viruses and aiding in destruction. They are associated with gamma globulin fractions of plasma protein and are produced by plasma cells. If you look at the structure and nature of immunoglobulins, all immunoglobulin molecules consist of two identical heavy chains and two identical light chains held together by disulfide linkage and non-covalent interaction. These immunoglobulins form a Y-shaped tetramer with a three-dimensional structure. The heavy chains of these immunoglobulins are linked to carbohydrates. Each immunoglobulin has two regions. A constant region, this portion remains relatively unchanged among immunoglobulins of the same classes. Other one is variable region. This portion is unique and allows the immunoglobulins to specifically bind to different antigens. The immunoterminal half of the light chain from the variable region, where the carboxyl terminal half from the constant region, as you can see in this figure. The amino acid sequence of the variable region in both light and heavy chains are responsible for the specific binding of immunoglobulins to antigens. Let's look at the types of immunoglobulins. There are 5 types of immunoglobulins found in humans: immunoglobulin M, immunoglobulin Z, immunoglobulin A, immunoglobulin E, and immunoglobulin D. Let's discuss one by one in a bit detail, starting with immunoglobulin M. Feature of immunoglobulin M. Immunoglobulin M is the largest immunoglobulin. Due to its large size, it cannot pass through blood vessel walls and thus restricted to the bloodstream. Immunoglobulin M is the first antibody produced in response to antigen and is the most effective against invading microorganisms. It can simultaneously bind to 5 antigens due to its structure. We'll see its structure in detail a bit later. Immunoglobulin M serves as the first-line defense and provide humoral immunity. Immunoglobulin M can bind very tightly to multiple antigens, and this binding induces the Fc region to activate the complement pathway, which leads to destruction of pathogen. Immunoglobulin M also activates macrophages to phagocytose pathogens. Immunoglobulin M can be splitted by enzyme papain into fragments. It produces 2 identical antigen-binding fragments, FAB, and 1 crystallizable fragment. If you see its structure, it exists as pentamer. The heavy chain is called mu chain and light chain is called kappa chain. It composed of 5 Y-shaped units held together by J joining polypeptide chain. Hope now you can easily answer this question correctly. Type your answer in the comment section below. Let's look at our next immunoglobulin, that is immunoglobulin Z. Features of immunoglobulin Z. Immunoglobulin Z is the most abundant class of immunoglobulins in the body. It composed of single Y-shaped unit and can easily traverse blood vessels. IgE is the only immunoglobulin that can cross the placenta, providing passive immunity to fetus. IgE triggers foreign cell destruction through complement system. It is mostly responsible for humoral immunity. The IgG class of antibodies is further divided into 4 subcategories: IgG1, IgG2, IgG3, and IgG4, which have gamma-1, gamma-2, gamma-T, and gamma-4 heavy chains respectively. Mother milks also contain IgG, providing passive immunity to newborn. If we see its structure, here oligosaccharides are attached to the protein backbone. It has gamma heavy chains and kappa or lambda light chain. It composed of 4 polypeptide chains held together by disulfide bond. N-terminal end of one heavy chain and its neighboring light chain cooperate to form antigen binding site. IgG is bivalent and can bind two antigens. Hope now you can easily solve this question. Type your answer in the comment section below. Let's look at our next immunoglobulin, that is immunoglobulin A. Features of immunoglobulin A. Immunoglobulin A is mostly found in body secretions such as saliva, tear, sweat, and milk. It is also present in intestines. In mucosal secretion, IgA is present as secretory IgA or SLGA, which has additional secretory components that protects it from degrading by enzymes. IgA is the most predominant antibody in colostrum, the first milk produced after childbirth. IgA binds to bacterial antigen present in the body surface and prevent them from entering the body, thus protecting body's surface from infections. If you see the structure of IgA, it can consist as monomer or a dimer connected by J chain. Heavy chain of IgA is alpha chain and light chain is either kappa or lambda chain. IgA has two antigen binding sites per monomer, allowing it to bind the antigens. Hope now you can easily solve these questions correctly. Let's look at our next immunoglobulins, that is immunoglobulin E. Feature of immunoglobulin E. Immunoglobulin E normally present in minute concentration in the blood. Immunoglobulin E levels are elevated in the individuals with allergies, as it is associated with body's allergic reactions. IgE present in tissues where, after binding to antigen, it stimulates mast cell to release various factors including histamines. Histamine causes blood vessel dilation and increase permeability, leading to symptoms of allergic reactions like itching, swelling, etc. IgE also activates eosinophils, a type of white blood cells, to fight parasites. If you see the structure, you can notice it exists as a monomer. The heavy chain of IgE is an epsilon chain and light chain is a kappa or a gamma chain, both chains held together by disulfide bonds. Fc regions of IgE bind to specific receptor on the surface of mast cell and basophils. Each IgE molecules has two antigen binding sites located at the FAB regions. Hope now you can easily answer this question correctly. Let's look at our next immunoglobulin, that is immunoglobulin D. The features of immunoglobulin D. Immunoglobulin D composed of single white cell unit and is present in low concentration in the circulation. It is primarily found on the surface of beta cell. There are no known specific function of immunoglobulin D, though it may act as beta cell receptor. If you see its structure, it exists as a monomer. The heavy chain is a delta chain and the light chain is a kappa or a gamma chain, both chains held together by disulfide bonds. It has two antigen binding sites located at the FAB region, that is the variable region of heavy and light chains. After looking at our armies, that is immunoglobulins, let's take a glance of another prospect of our immune system, that is hyperactive immune system and autoimmune diseases. In general, the immune system is self-tolerant, meaning it doesn't attack the body's own cell or proteins. However, in certain conditions, the immune system fails to differentiate between self and non-self, leading to an attack on body's own cells or tissue. This phenomenon is referred as autoimmunity, and the related diseases are called autoimmune diseases or disorders. You should be able to solve this question from previous slide. Let's look at some types of autoimmune diseases. First one is rheumatoid arthritis. In rheumatoid arthritis, the immune system targets the lining of joints, leading to inflammations, pain, swelling, and eventually joint damages. The next one is systemic lupus erythematosus. It is a chronic disease where immune system can attack various body systems including the skin, kidney, heart, lungs, and brain. Symptoms include fatigue, joint pain, skin rashes, and organ damage. The next is type 1 diabetes. It is the autoimmune disorder destroys insulin-producing beta cells of pancreas, leading to an inability to regulate blood sugar level. The next is multiple sclerosis. In this disease, the immune system attacks the protective covering, that's myelin, of nerve fibers in the central nervous system, leading to impaired communication between brain and the body, resulting to muscle weakness, coordinations of tissues, and vision problem. Next is Graves' disease, where the immune system targets the thyroid gland, causing it to overproducing thyroid hormone, leading to symptoms like weight loss, anxiety, and rapid heartbeat. Next one is Hashimoto's thyroiditis. The immune system attacks the thyroid gland, resulting reduced thyroid hormone production, causing fatigue, weight gain, and cold intolerance. Next one is psoriasis, where the immune system causes skin cell to multiply too quickly, leading to scaly, inflamed patches of skin. Next one is celiac disease. Celiac disease is an autoimmune reaction to gluten, a protein found in wheat, barley, and rye that damages small intestine, leading to digestive issues and nutrient malabsorption. The next one is myasthenia gravis, where the immune system interferes with communication between nerves and muscle, causing to muscle weakness and fatigue. The next one is inflammatory bowel disease, that is IBD, which includes diseases like Crohn's disease and ulcerative colitis, where the immune system attacks the lining of digestive tract, causing inflammation, pain, and digestive problems. These were the most important examples of autoimmune disease. Now solve this question and drop your answer in the comment section below. With this question, let's wrap up today's session. Let me know how this session was. You can also drop your doubts and valuable suggestions in the comment section below. Hope you liked the discussion and thank you for watching.